Good evening. My name is Andrew Goodliff. I'm Associate Dean of the Graduate School. On behalf of the grad school, I want to welcome you all to the 2024 Three Minute Thesis Finals. So this competition is very much a highlight of the year for the graduate school. This competition is a research competition and creative activity communication competition. The participants are challenged to present their thesis or dissertation or creative work in 3 minutes in such a way that a non-expert audience can understand the importance and impact of their work.
3MT is a training opportunity disguised as a competition. Students learn to present their research topics to a broad audience in ways that really emphasize the life-changing importance and potential impact of the projects that they're devoting years of their lives to carrying out.
The goal is to train students to be able to distill their complex research down to 3 minutes and down to something that a regular, educated person off the street can understand.
They're given these very strict parameters that they have to follow, and it really forces them to think about their work in a really sort of focused and articulate way.
The reason the 3MT competition focuses on 3 minutes and 1 slide is to help students get directly to the heart of the why. What we train them to do is to grab the audience's attention with this big picture, gauge the interest level of their listener, and then they can go into more detail.
These are students who are doing things that relate to the everyday person on the street, be they anywhere in Alabama. And I want these people to go away with saying, hey, no, I can see the applicability of this. This is good. This is actually furthering technology. This is helping me.
We're very grateful to these judges for making the time to help us tonight. As I introduce each judge, ordered alphabetically by last name, please raise your hand. Hold applause for when I've finished introducing the judges. So, alphabetically, first is Dr. Susan Carvalho, Associate Provost and Dean of the Graduate School from the University of Alabama. Second, Dr. Kathy Randall, Chair of Pettus Holdings, LLC. Third, Dr. Christine Taylor, Vice President and Associate Provost for Opportunities, Connections, and Success at the University of Alabama. Last but not least, Dr. Stuart Uzdan, Dean, College of Human and Environmental Sciences. Thank you again for the judges for making the time tonight. Now, a note on how the presentations will progress. Each participant will give their presentation, 3 minutes. This will be followed by approximately 60 seconds while the judges tabulate their results. So there'll be somewhat of an awkward pause, mostly awkward for me standing up here, between each of the presentations. Now, the reason that we are actually here, our finalist presentations. I really want to thank our presenters. It takes a huge amount of courage to get up and participate in this competition. This is not something they have to do. This is something they have chosen to do. And this is something they will remember for the rest of their lives going through this process. It is an amazing thing. And again, this is a really important skill. All right. I want to ask our first participant to step forward. Our first participant is Samantha Burke from College of Communication Information Sciences. The title of Samantha's talk is New Ways to Study Grief, Public Memorials in the United States. Thank you, Samantha.
In almost every classroom in America, you can find a sheet of paper with 3 simple instructions. Run, hide, or fight. These explain to students and educators what they should do in the event of an active shooter. My generation and younger has never known an education without an underlying fear of gun violence. But talking about school shootings makes people uncomfortable. As such, the research in this space is limited, primarily existing within psychology and criminal justice. My research aims to bring the study of school shootings into communication. I do this specifically by looking at the permanent memorials that are built in the aftermath of these tragedies. I'm interested in how they both physically function, as well as the larger conversations that they create. I do this through a multitude of ways. First, by using the theory of space and place, which explains how spaces and identity are connected, as well as places and persuasion. Secondly, the theory of grievability, which explains how some lives are valued more than others, and how this phenomenon persists in death as well as life. I use these two lenses to analyze all of the communication that surrounds these memorials, from proposed architectural plans to city council meetings where they're approved. Also social media posts and news articles that are written about these memorials after they open. Let's take, for example, the Sandy Hook Memorial that we see here. The gravel path that converges upon a cool pool of water. The single sycamore tree that sits in the middle, whose leaves catch the sunlight. And the smooth granite that encases the pool. This memorial was built to create an environment of peace. My analysis has shown that memorials like these function in a multitude of ways. First, they provide a physical location for people to gather, to remember, and to mourn. But secondly, they create a conversation about grief. These allow people to get together and speak. These memorials provide hope for families who've experienced unimaginable loss, for communities who are healing in the aftermath of tragedy, and hope for all of us that we are never truly alone in our grief. My research aims to do the same, provide hope in the emptiness of tragedy. Thank you.
Again, a short pause while the judges tabulate their scores. Our next speaker is Adriana Riveros. Adriana is from the Department of Civil, Construction, and Environmental Engineering. The title of Adriana's talk is Removing Toxic Selenium from Water Using Innovative Electric Biotechnology. Thank you.
Imagine taking a sip of water and feeling uncertain if the water will refresh you or actually harm to you. For millions of people, this isn't just a feeling. Water contamination is a daily reality. One pollutant that is affecting over 40 million people across the 5 continents is selenium, specifically in its toxic form of selenate. Selenate in water is devastating, as has wiped out an entire fish population in North Carolina. But it's also harmful for humans' health. It can cause liver damage, reproductive failure, and even cancer. Today, I'll talk about how my research approached this problem and uses a novel combination of 2 technologies: flow electrode, capacitive deionization, and bioelectrochemical systems, and how these 2 work together to treat selenium in water for the first time ever. Let's start with flow electrode capacitive deionization. This technology uses electricity to remove and attract selenate out of the water. As you can see, selenate has a negative charge, and we know that negative and positive attracts, so we will attract it to a positively charged electrode. Essentially, it's like a magnet that is pulling out the selenate out of the water, making it cleaner. But it doesn't stop there. If you think about it, even though we remove the selenate out of the water, it still remains in its toxic form of selenate. To convert toxic selenate to non-toxic selenium, we use a special bacteria. We call it electroactive bacteria. Why electroactive? Because they can use electricity and electrons to transform selenate to non-toxic selenium. Basically, we're feeding the bacteria with harmful and toxic selenate, and the bacteria is eating it, transforming it, and giving us back non-toxic selenium. Amazing, if you ask me. This process, it is called bioelectrochemical systems. As bio refers to the bacteria, and electrochemical to the use of electricity to transform this toxic selenate to non-toxic selenium. Gladly, I can say that for the first time, I was able to prove that the combination of these two technologies can effectively remove selenate out of the water, all by taking advantage of electricity, and hopefully making water safer for everyone. Thank you.
Our next speaker is Amy Matthews. Amy is from the School of Accountancy. The title of Amy's talk is Job Stressors and Career Commitment in Early Career Auditors' Work Experiences. Thank you.
Imagine starting your career eager to make a difference and perform at your very best. You're equipped with tools and resources to help boost productivity, promote flexibility, and make work more fulfilling. Professionals today are able to work remotely, coordinate with individuals across different time zones, and are encouraged to use innovative methods to help boost productivity and perform their work more effectively. Yet ironically, these very practices intended to simplify work can often cause it to be more demanding, leading to unexpected stress, especially for those just starting out. My research focuses on identifying specific factors that cause stress in the work lives of early career auditors and examining how these stressors affect their well-being and commitment to the profession. Studies find that when employees are not able to mentally disconnect from work, this causes higher stress and makes them more likely to feel dissatisfied, perform poorly, and even consider quitting. On the other hand, when they are able to disconnect and switch off at the end of the day, this helps to reduce stress by promoting recovery and therefore improving overall well-being. I surveyed early career auditors about their experiences of having to perform job duties outside of established hours, the factors that contributed to these experiences, and their ability to disconnect. This helped me pinpoint key stressors and understand how these experiences shape their view of the profession. The main stressors identified in my survey were feeling pressured to continue working and a constant demand to stay connected through online messaging and evening and weekend hours. Many auditors found it difficult to truly switch off at the end of the day and struggled to disconnect from work. Despite these challenges, though, my initial findings find that early career auditors have an overall positive view of the profession and the work that they are performing. By capturing their real experiences, my study highlights the unique challenges that early career auditors face. With high turnover in accounting, my findings emphasize the need for strategies to help auditors manage stress and disconnect after work. My research aims to improve the overall work experience for early career auditors, thus helping to strengthen and stabilize the future of the profession. Thank you.
Our next speaker is Kyle Brolt. Kyle is from the Department of Biological Sciences. The title of Kyle's talk is Tiny Insects, Big Impact, DNA Analysis of Caddisfly Diets. Thank you, Kyle.
Raise your hand if you ate meat today. Fruit, vegetable. All right, that was easy. It's not hard to figure out what humans eat because we can just ask. But biologists can't do the same thing for animals. Sure, sometimes it's easy. We can see a lion devour a gazelle. But something like an aquatic insect that lives in freshwater ecosystems, this can be a little bit more challenging to answer. Yet biologists are obligated to do so. Because if we want to conserve and protect any organism or ecosystem, it is a prerequisite that we understand their food web dynamics, or who eats who and what eats what. In the case of aquatic insects, this is particularly important because they form the foundation of our freshwater ecosystems. And as our waterways become warmer, drier, and more polluted, we need to know how insect diets change over time so we know how they're responding to these negative stressors. That way we can make informed conservation decisions. But unfortunately, it can be really hard to evaluate what aquatic insects are eating. Take, for example, the Pycnosphaera caddisfly. The Pycnosphaera caddisfly falls into a feeding regime that aquatic biologists call shredding insects. This means that they eat dead leaves that fall into the water. And these leaves are going to be colonized by a diverse assemblage of bacteria and fungus, both of which are vital for any shredding insect's nutrition and health. But that's the major extent that we understand about the Pycnosphaera caddisfly and honestly other shredding insect diets. The problem is, if we want to know what they're eating, we have to break open their stomach contents and look under a microscope. But us biologists have a really hard time knowing what species of plant or fungus we're looking at because it's been eaten, it's shredded up, and it's half digested. So my solution is to use whole genome shotgun sequencing on the Pycnosphaera caddisfly's stomach contents. And for me, my workflow for the whole genome shotgun sequencing can be broken down into 3 major steps. First of all, I'm going to utilize some scientific wizardry to isolate the DNA from the plant, bacterial, and fungal cells that I find in the Pycnosphaera caddisfly stomach. Secondly, I will do DNA sequencing to get the unique genetic code for each organism present in their guts. Finally, I will compare the genetic codes I've compiled to the genetic codes of known species and match them. So this genetic code is this species, and this genetic code is this species. My hope is that my research can be used by other aquatic biologists in the future so we can better understand these insect diets, who knows, maybe even better than we understand the diet of a lion. Thank you.
Our next speaker is Paramita Sarkar. Paramita is from the Department of Chemical and Biological Engineering. The title of Paramita's talk is Making a Brain-like Environment to Understand Drug Impact on Breast Cancer. Thank you.
Have you ever tried solving a puzzle only to realize that the pieces needed to complete it are missing? Frustrating, isn't it? Now imagine that this puzzle is someone's health and the pieces missing are critical to saving their life. Breast cancer affects millions of women worldwide. While many fight and overcome it, for some, it can spread to other parts of the body. For example, the bone, brain, and lung. If it reaches the brain, it becomes one of the most devastating challenges that they can face because treatments that work elsewhere in the body often fail in the brain. And the reality is heartbreaking because 90% of these women lose their battle within the first 1 to 2 years. It's like we're fighting with our hands tied, unable to find these missing pieces. My PhD thesis is all about finding those missing pieces. To solve this challenge, first we need to understand how breast cancer cells behave in the brain. But since we cannot just open a human brain and look inside so easily, I have created a brain-like environment in the lab. I made a special substance that resembles a jello, except this version is not edible. It is made using hyaluronic acid, which is a key building block of the brain's natural environment. It allows my jello to be able to mimic the softness and feel of the human brain tissue. But it gets even more exciting from here. I can actually modify this jello by adding specific components that are present in the brain, like different cells and proteins. And by adding these components, one at a time, I'm being able to figure out their individual effects on these cancer cells. But this is not where I stop. After setting up this brain-like environment, I then introduce drug treatments. Then I observe and I ask the important questions. Do the drug stop the breast tumor from growing or change how they behave? By testing the drugs in different brain-like settings, I am able to figure out which treatment might be the most effective in the real brain. And I can also uncover why the cancer cells behave the way they do and potentially how to stop them. Every experiment is like finding those missing pieces and adding it back to the puzzle. And at the end of the day, it's about more than just science. It's about hope for countless women who have fought, who are still fighting, and those we love. And every discovery we make takes us a step closer to a world where breast cancer, even in the brain, is not a death sentence, but a battle we know how to win. Thank you.
Our next speaker is Shabnam Salehi. Shabnam is from the Department of Information, Statistics, and Management Science. The title of her talk is Can We Incentivize Supermarkets to Reduce Waste and Hunger by Donating Food? Thank you.
Did you know that supermarkets in the United States throw away over 16 billion pounds of food each year? Meanwhile, over 47 million people experience food insecurity in this country. In a rich country like United States, these two issues, food insecurity and food waste, shouldn't coexist. The food bank supply chain, where food banks receive donations from supermarkets and distribute them to food insecure households, addresses both food waste and food insecurity. However, food banks face main challenges when it comes to perishable products like milk. They prefer such perishables to arrive early enough to distribute them before they spoil. So they prefer perishables with longer shelf lives. However, intuition tells us that supermarkets should donate perishables at the very last minute, very close to their expiration, for a chance to sell them at full price. And this highlights a fundamental conflict between supermarkets and food banks. And here is where my research comes in. Our research adopts supermarkets' perspective because we want to incentivize supermarkets to donate perishables earlier in their life cycles. We formulate a mathematical model that describes the interaction between different stakeholders in the food bank supply chain. We apply optimization techniques that tell supermarkets the optimal timing and also quantity of donations. We simulate a robust set of operating conditions, and we find that donating perishables earlier in their life cycles not only maximizes supermarkets' profit, but also reduces waste by over 70% compared to last-minute donations. And this result is counterintuitive because previous studies have shown that societal objectives like waste reduction and profit rarely align. However, our research offers a win-win scenario where supermarkets can reduce waste without sacrificing their profit. Therefore, alleviating food waste and food insecurity is not just about what individuals like you and me can do. Our research provides financial incentives for supermarkets to step up and play a significant role in the fight against food insecurity and food waste. Thank you.
The next talk is by Braden Hall. Braden is from the Department of Psychology. The title of Braden's talk is Flirting with Faculty.
College students flirt. They flirt like a lot. They flirt with a lot of different people in a lot of different places for a lot of different reasons. They flirt in bars to get free drinks, at the library to just make friends, or at the coffee shop to get the barista's number. But college students flirt in their classrooms too. And they don't just flirt with their classmates, they flirt with their professors too. Now, this might come as a shock to all of you and leave many higher education administrators clutching their pearls, wondering how many of their well-respected students are sharing seductive glances with their professors. Well, to examine whether this is the case, we surveyed over 230 students from an unnamed university and asked them how often they engaged in 24 different flirting behaviors, like wearing a tight shirt to class or twirling their hair while their professor lectures. And what we found is that we also asked them how often they thought their friends engaged in all of these behaviors, and we asked them how appropriate they found these behaviors to be. Now, you'll be happy to know that almost all of our participants reported never engaging in any of these behaviors, that they didn't think their peers engaged in these behaviors, and that these behaviors were wholly inappropriate. But don't worry, it gets spicier. Because 43% of our sample also reported that they believed a student could raise their grade in a class by flirting with their professor. But what makes a student think that biting their pen could score them an A? Well, to get to know our participants a little more and to examine their Casanova strategies, we also examined their personality traits, one of which was an indicator of grandiose narcissism. Now, for the non-psychologists in the room, grandiose narcissism is a personality trait that's characterized by an inflated sense of self and unreasonable entitlement. But it's also characterized by charm, confidence, and social boldness, which makes someone a pretty good flirt. And what we found is that participants higher in grandiose narcissism were more likely to believe that they could flirt their way into an A. They were also more likely to engage in every single one of the behaviors that we measured, likely because they flirt in every other area of their life, so why not flirt with my professor while I'm at it? We also found that they were more likely to say that their friends engaged in these behaviors, and if I'm a grandiose narcissist, why isn't everyone doing what I'm doing? We also find that they think these behaviors are more appropriate, because if I'm a grandiose narcissist, everything I do is appropriate. But all in all, while professors are unlikely to walk into a room full of clamoring narcissists desperate to flirt with them, they are likely to encounter a student every now and then that, because of their personality, thinks that a tight shirt, a dirty joke, or a well-placed compliment might just make its way into the grade book. Thank you.
Our next speaker is Kabarat Lola. Sorry there. Kabarat is from the Department of Translational Science and Medicine. The title of her talk is Treatments Aim for Precision, but Lupus Medicines Often Miss the Mark. Thank you.
In our daily lives, we are constantly exposed to various disease-causing microorganisms. Fortunately, we have an immune system that works round the clock to protect us. Our immune system identifies and combats invaders such as bacteria and viruses. But just like every other system in the body, the immune system can sometimes malfunction. In a situation where the immune system thinks there are invaders, but there are actually no invader, it's a false alarm, and it triggers a series of immune responses that accumulate in an autoimmune disease called lupus. Currently, 1 in 2,000 Americans suffer from this disease. The logical goal of treatment is to suppress these erroneous immune responses using a class of drug called immunosuppressants. However, immunosuppressants are typically not the first line of treatment for lupus. And this is primarily because this drug affects multiple systems and organs in our body, leading to unintended side effects and reduced effectiveness. While drugs are prescribed to treat specific conditions, oftentimes they can produce unintended results. Is there anyone in this room tonight who has ever wondered why missiles, even though they are fired from very far places, get to hit specific intended targets? I will let you in on the trade secret. Missiles are equipped with extremely accurate guidance systems, such as electronic sensors and GPS that guide them to their target. Now, imagine if immunosuppressants can be equipped with such accurate guidance system to ensure their direct delivery to the immune system without affecting the rest of the body. That would be a groundbreaking discovery. I believe I'm making significant strides in advancing this field. My research focuses on the targeted delivery of immunosuppressants to the immune system using accurate guidance system. I have encapsulated an immunosuppressant in a nanoparticle, and I attach a ligand that functions like a GPS. So just as the GPS guides missiles to their targets, my ligand guides immunosuppressants to identify and interact with immune cells, which are the key players in lupus. I have tested this treatment in 2 mouse models of lupus, and I have validated the hypothesis that targeting immunosuppressants to the immune system outperforms the traditional use of this drug in the management of lupus. The success of my research will positively impact the lives of millions of people who are currently living with the disease. Roll Tide.
Our next speaker is Henry Mouser. Henry is from the Department of Biological Sciences. The title of Henry's talk is A Microbial Murder Mystery, How the Blood-Brain Barrier Fails During Meningitis. Thank you, Henry.
In a murder mystery, the greatest threat is often the one you least suspect. Pneumococcus, or pneumo, the bacterium depicted in green, is an unassuming microbe. It typically lives in the nasal passages of humans without ever causing disease. You never suspect then that this same microbe accounts for nearly 40,000 deaths per year due to an infection known as meningitis. Meningitis occurs when bacteria in the bloodstream cross into the brain and cause overwhelming inflammation, which often proves fatal. Now, in actuality, very few bacteria are able to get into the brain due to a structure known as the blood-brain barrier. The blood-brain barrier is a network of specialized cells that separates the blood from the brain. You can think of these cells like bricks that form the wall that is the blood-brain barrier. And typically, the barrier is highly effective at keeping dangerous pathogens and toxins outside of the brain. However, we know that pneumococcus is able to breach the blood-brain barrier and access the brain, but how it does so is still largely a mystery. One of the greatest obstacles to solving this mystery is simply our ability to understand this disease. In natural settings, pneumo only infects humans, but scientists clearly can't infect and dissect humans to study meningitis. So we're faced with the dilemma of if we can't study it, we won't understand it. And if we don't understand it, we cannot treat it. My work is addressing this dilemma. My lab and I are able to take non-embryonic stem cells from human donors and over a 14-day process, differentiate them into cells that express all of the key hallmarks of the blood-brain barrier. This cell model has been validated and utilized in numerous publications. I'm not the first to use it, but no one has ever used this model to study pneumococcal meningitis. So that's exactly what I did. You can actually see these cells shown via a technique called immunofluorescent microscopy. This allows us to visualize specific cellular structures. You can see the nuclei of each cell shown in blue, and in red is a protein called ZO1. Now, if the barrier is a wall and the cells are the bricks, then ZO1 is the mortar that holds everything together. It helps create that impenetrable barrier so that nothing can slip through. And it's doing a great job of it in our healthy, uninfected condition. But when I infected with pneumococcus, I observed the loss or degradation of ZO1. This suggests that pneumo is destroying ZO1 as a means of creating a chink in the wall, making a hole in the barrier and slipping into the brain to cause meningitis. This is just one of the many discoveries I've been making, and while the mystery is still far from solved, my work is bringing us closer and closer to our goal of understanding, treating, and eventually beating this disease. Thank you.
Our next speaker is Raj Kumar Shah. Raj Kumar is from the Department of Chemical and Biological Engineering. The title of Raj Kumar's talk is Enhancing Water Filters for Urea Removal from Wastewater. Thank you.
Urea, one of the most widely used fertilizer at every nook and corner of the globe for crop production, with exceeding the production limit of 180 million metric ton per annum. Having said that, 55% of all the fertilizer is only depend on urea, which means the demand of urea has been exponentially skyrocketed over the past couple of decades. It's not only useful as a fertilizer, but it can also be useful as a cosmetics, healthcare, and many more applications. However, according to Environmental Protection Agency, this urea can leach into the freshwater body sources, such as ponds, lakes, and river, and could cause eutrophication. For instance, a few weeks ago, I was traveling to Florida and I saw some of the ponds covered by this green layer. This layer is adversely affecting the aquatic life and flora and fauna residing into these freshwater body sources. It could create the deficiency in oxygen and could block the sunlight, ultimately damaging the ecosystem. So what's the solution? So the current wastewater treatment plants are able to remove urea using biological treatment, but it requires humongous amount of energy and space, which is not something rational in this 21st century where we talk about the concepts like circular economy and sustainable development. Therefore, there is an urgent need to develop an innovative solution to mitigate this problem. As a prevalent quote said, Every cloud has silver lining. Our nature has already resolved this issue. Yes, did you know our human kidney can filter 180 liters of blood per day, including contaminant like urea, without requiring any energy and space? There my research comes. So our kidney filters urea using specialized protein called aquaproteins, which are similar as a soap-like structure, a soap that we use in our day-to-day life to clean our hands, to clean our dishes, to do a laundry. So what I do in my research, I try to incorporate these soap-like molecules in order to make artificial water filters. To imagine a water filter, you can have a coffee filter that we use to filter our coffee. But here it becomes efficient enough that can remove urea, which requires 40% less energy as compared to conventional wastewater treatment plant. In addition to that, this water filter can also be useful for the 2 million kidney patients worldwide who are undergoing the treatment of dialysis, where they can use this water filter as a portable device attached with them, which requires less energy, time, and cost. Thus, in a nutshell, my research provides a solid platform to fight against the ecosystem through sustainable development as well as healthcare upgradation. Thank you for listening to me.
Our next speaker is Faye Bramblepelt. Faye is from the Department of Biological Sciences. The title of Faye's talk is Genes Help Us Understand Our World. How can we make studying them more accessible? Thank you.
Can I tell you guys the thing that made me fall in love with biology? How much of the DNA in your body do you think is genes? Genes being the things that have the instructions that help make tissues, our body, they make us who we are. Do you think maybe like 50% of DNA, 70% of DNA? It's 2%. Every single bit of what makes you who you are is just 2% of all the DNA in your body. So what's the rest of it, right? Most of it is repetitive elements. All this junk that doesn't actually contribute to making the proteins that make you who you are. Those repetitive elements have these little carbons on them that help pack them up and keep them out of the way. Those marks, called methylation marks, are clues that I think we can use. I'm testing special enzymes that work like molecular scissors. They cut DNA right at those special methylation marks and leave behind anything that's not marked. What I've done is I've taken DNA and I've treated it with that enzyme. Hopefully, fingers crossed, those non-genic regions would get chopped up really fine, but they leave behind big chunks for the genes. Then I have a problem. I don't really know what's in my sample until I sequence it. So I'm forced to sift it out. Sift out all the small, tiny bits that I think are not going to be very important and keep behind those big chunks that, fingers crossed, are going to be genes. It's kind of like panning for gold. When you pan for gold, you take this giant chunk of information and shift out all the small bits and keep behind those gold nuggets. For me, those gold nuggets are the genes. Those genes, so far, my work has been able to produce an over 300% increase in gene space coverage compared to a control. That's incredible, because when we care about genetics, to better understand how a crop field can be resilient towards change or how it can produce more fruit, when we care about something like that, we can spend $50 on one individual and only get back $2 worth of genes. That's horrible. By increasing the amount of genes we get back per sample, I'm helping make it so we can spend a lot less money to get a lot more done. And that matters to me because genes are really important. I'm hoping we can use my technique to better understand historical samples to see changes across the global scale, or it could even have implications for human genetic health and testing. But ultimately, genetics is really cool and really expensive. So making it a little bit cheaper helps make it more accessible to all kinds of scientists, which is why I think my work is going to help change the world. Thank you guys so much.
Our next speaker is Pawani Khadka. Pawani is from the Department of Economics, Finance, and Legal Studies. The title of Pawani's talk tonight is Strategically Mapping Your Emotions. Thank you.
I'm here to talk to you about the economics of manipulation. How easy do you think is it to manipulate somebody into trusting you? Businessmen, charitable institutions, your friends with the most amazing ideas could all be trying to gain your trust so you can invest in them or a cause close to them. In my research project, I use a simple investment game, apply some modifications to it, and see whether one can use emotion manipulation as a strategy to gain trust. The basic structure of the investment game is as such. There are two randomly and anonymously matched players, both given the same amount of money. One of them, say player 1, is asked to send some or all of that money to the other player, say player 2. The catch here is any amount sent is tripled before the second player receives it. You can already see there is an incentive for player 2 to get player 1 to trust and send more. However, there's no interaction between these players except the give and take. What I uniquely introduce here is allowing player 2 to potentially manipulate player 1's emotions. Induced emotions have been shown in literature to cause behavior that's different from theoretical predictions. But the impact of emotions, specifically on trust behavior, is relatively understudied. So in my research, I allow player 2, now the manipulator, to choose a video, a happy and exciting or a neutral one, to show to player 1, now the truster, before they make that send decision. When given that choice, 96% of player 2s chose either a happy or an exciting video over the neutral one. But we find no statistically significant differences in the amount sent just based on the video type. One might think here it's probably because my videos didn't work. We used facial recognition software on over 400 participants recruited at the UA Tide Lab and confirmed that each of the videos do have the intended emotional impact. However, these emotions die out so fast that it didn't matter what video was watched by the time they made their send decision. Player 1s were shown these videos in one of two ways. It was either a computer randomizing the video for them, or it was player 2, the manipulator, choosing a video for them. And when player 1 knows it's player 2 that's chosen the video for them, they sent on average 13% less than they did in the other case. So, what do we learn? It's that the type of emotion manipulation tool is not as important. But if people sense manipulation, they do become protective and trustless. So, if you're that business person or the friend trying to raise money for your favorite puppy shelter, make sure your potential donors cannot identify your method of persuasion.
Our next speaker is Godwin Sunday. Godwin is from the Department of Geological Sciences. The title of Godwin's talk tonight is Rescuing Our Rivers from Abandoned Coal Mines. Thank you.
Hello, everyone. I'll be discussing a very critical issue today, how abandoned mines has affected the quality of our water and the environment, and the necessary steps taken to fix it. But before then, I want us to imagine a very beautiful and a pristine river flowing through a lush landscape, supporting plants, fishes, and aquatic life, a source of clean water to the nearby community. But then, mining happens, and everything drastically changes for bad. This slide will show you the journey of a watershed on how clean and untouched, pristine water goes from being clean to being impacted by pollution, and even how it might look like after it has been restored. The culprit here is coal mining. When mining waste are being exposed to the atmosphere, it tends to react with water and air, forming what we call acid mine drainage. This makes the watershed highly acidic. Thus, it dissolves whatever surrounding materials it comes in contact with, thereby mobilizing some toxic substances into the environment. Example of those toxic substances could be aluminum, manganese, iron, and nickel. My research has revealed that these toxic substances are present in concentrations well above the safe limit in some areas of this watershed. This is dangerously harmful because aluminum and iron can be deadly to fishes and aquatic life when they are being high in concentration, while manganese and nickel are being known to cause certain neurological and developmental issues when they are being consumed in drinking water. To address these concerns, I collect water samples from Brookwood, a community in Tuscaloosa that has been known for coal mining since the 1800s. I do this in collaboration with the Geological Survey of Alabama. I test these water samples to track the acidity and metal concentrations. I gather and send this data to the governmental organization known as AMLA, who sponsors my research. This information would guide them into carrying out effective remediation through targeting the most affected areas. As someone that cares deeply about environmental health, my ultimate goal is to see that my research turns into real action for cleaner water and a healthier ecosystem. Thank you for your time.
Our next speaker is Abiodun Wahab. Abiodun is from the Department of Translational Science and Medicine. The title of her talk tonight is Navigating Cancer Chemotherapy, the Good, the Bad, and the Transformative. Thank you.
Cancer is a devastating disease. It is one of the most leading causes of death worldwide. The National Cancer Institute estimated that by 2040, over 20 million new cases of cancer will be diagnosed, and over 15 million people will die from this disease. For many cancer patients, chemotherapy is an essential part of their treatment journey. But unfortunately, chemotherapy drugs doesn't just target cancer cells. They also kill healthy ones, leading to severe side effects. You see cancer patients losing their hair, having multiple organ failure, and different complications from their treatment. Oftentimes, clinicians have to withdraw the use of chemotherapy agents to allow cancer patients some time to recover. Now, while they are recovering, cancer cells continue to grow unchecked. They become resistant to the initial drug that are being used to treat them. Cancer patients become sicker, and their overall quality of life is being compromised. Take a second to imagine a world where cancer patients can effectively fight their disease while avoiding the severe side effects that comes from their treatment. That's exactly the world my research aims to provide. My research is focused on a small natural compound called urolithin A. It is commonly found in fruits that we eat. It has shown potential to target cancer cells and also mitigate side effects from chemotherapy. But the challenge is, the amount that we take from fruit is still little to be effective. Also, when we take it in as supplement, it gets metabolized real quick before it gets to the target organ to even reverse any effect. In about 3 hours, it is gone. So to address this issue, I reformulated urolithin A using advanced nanomaterials. Now, these nanomaterials are tiny nanoparticles that hold it in place long enough to get to the target site, reverses every side effect from chemotherapy. I have tested this new formulation on human cells and different animal models, and I found that not only does it reverse every side effect from chemotherapy, but it also makes sure that it stays enough to be used for reversing side effect. It's exciting to note that this new formulation can be a long-term addition to standard chemotherapy treatment that allows cancer patients to continue their treatment without interruption, reverses side effects from chemotherapy, and offer them better chances in their fight against cancer. In summary, my research aims to make cancer treatment more safer, more effective, allowing cancer patients not just to survive, but to truly thrive. Thank you.
Our final speaker tonight is Divya Baghel. Divya is from the Department of Chemistry and Biochemistry. The title of Divya's talk tonight is there really a link between Alzheimer's disease and type 2 diabetes? Thank you.
Imagine a world where your most precious memories, laughter of your loved ones, scent of your childhood home slips away, leaving all emptiness. This is the reality for millions suffering from Alzheimer's. It erases the essence of who someone is. In United States itself, the count is expected to reach 8 million by 2025, which is just in few months. And sadly, there's no cure yet. Studies also estimate that up to 81% people living with Alzheimer's also have type 2 diabetes, a condition that affects how your body manages sugar. During this, there is an excess sugar buildup in the body that can affect multiple organs, including heart and the kidneys. At first glance, these two seems unrelated, one affecting the brain while other the kidneys. But there's a catch. Recent studies have shown that people suffering from type 2 diabetes are more prone to develop Alzheimer's. Now, this odd connection has raised a big question. Are these two related somehow at molecular level? Though, evidence points towards the connection, but no one really has shown how type 2 diabetes can lead to Alzheimer's. And that's where my research began, to unlock and understand that molecular level connection between these two diseases. For Alzheimer's, one of the proteins called amyloid beta start accumulating in the brain, forming long strands of fibrils. Let's imagine your favorite hairstyle. Now, in case of type 2 diabetes, another protein called amylin starts accumulating and it forms its own fibrils in pancreas. Let's say your second favorite hairstyle. Now here's when things get interesting. When we mix these two proteins in the lab, these two do not stay in their own braid. Instead, they mix with each other, forming some new blended structure. To analyze these mixed fibrils, I use atomic force microscopy integrated with infrared spectroscopy. In simple words, just imagine AFM as a super sensitive fingertip that can feel the texture of different hairstyles. But again, feeling the shape isn't enough. We want to know what these braids are actually made of. Do they really contain both protein A and B, or just one of it? That's where IR steps in. It acts like a molecular detective, differentiating between, let's say, curly, wavy, or straight hair. So, in our lab, this combination of AFM with IR allows us to look at the mixed protein aggregates in a way that no one else has. AFM-IR acts like a key, unlocking that mysterious door with the hope of finding insights that could bring us closer to the cure, eventually helping millions suffering from these devastating conditions. Because when silent enemies forces against us, it's time to unite our knowledge and fight back for the sake of, thank you.
All right, if you can give everyone a round of applause again, I think we can all agree those were amazing. All right, now it's time for the audience to participate. You have your People's Choice ballot, and I want you to fill that out. And then on the way out, you're going to hand that to one of the ambassadors who has a basket. So give them your People's Choice ballot. Now, while you're doing that, we are going to ask the judges to adjourn. So if we can have the judges adjourn first, and one of our people will take them to the room.
And I do want to say, I mean, 90% of these could win regionals.
And great content.
Yeah.
Several of them really did an excellent job at describing their research.
Well, it is my honor to be able to announce the winners. But I have to first start by saying how flummoxed the judges were as we tried to differentiate between these amazing presentations. And one of the judges actually started the conversation by saying, what if we do just give them all perfect scores? What happens then? So the conversation went from there, but it really, we want to congratulate all of the presenters for amazing us with their command of their research and their ability to translate it to non-expert audiences. And even more than that, a layer of passion about the impact of what you're doing that was communicated and felt by all of us, that added to the difficulty of ranking anything. So, and then you look at the range of topics and how you can compare the apples and oranges, and it made the task extremely difficult. So we just want to start by hoping that all of you feel the same pride in what you've done as we felt in hearing it. So, that said, I am here with Solomon Ajasa. He is a PhD student and president of the Graduate Student Association, which is a major supporter and contributor to this event. Thank you for being here, Solomon. And he will hand out the envelopes. So I'm going to start by announcing the 4th place winner, and I'll go up from there. If I call your name, please step forward and take the envelope and stay there. So, in 4th place, Samantha Burke from Communication and Information Sciences. Thank you. Our presentation was New Ways to Study Grief, Public Memorials in the United States. Third place, Henry Mauser. Biological Sciences. And Henry's presentation, as a reminder, a microbial murder mystery, how the blood-brain barrier fails during meningitis. Second place winner, Paramita Sarkar from Chemical and Biological Engineering. And her presentation, Making a Brain-like Environment to Understand Drug Impact on Breast Cancer. The People's Choice winner, Kyle Bro from Biological Sciences. And his title was Tiny Insects, Big Impact, DNA Analysis of Caddisfly Diets. And if I could remember that word that was before caddisfly, I would include it. None of us managed to capture that. Thank you, I still don't know. And the first place winner, Kyle Bro. We know you all 15 will go very, very far. Thanks to all of you and your professors and your colleagues who are here in the audience. I know they also share the pride in what you've done. I just want to say a few more words. I want to thank my fellow judges, Drs. Kathy Randall, Chris Taylor, and Stuart Ustan, for your good work. Andy Goodliff always does an amazing job with all of the lead up to this presentation. And when he wears his dinosaur tie, you know you've really arrived. So you can see that this was a dinosaur tie night. Thanks also to Deanna, Tierney, Patrick, our timer, and the amazing team that pull all of this together and make it look easy, and all of our student ushers who helped us find our way in and hopefully will help us find our way out. And a special thanks to the UA Digital Media Center for allowing us to use the space, and for the audience who comes out to support these amazing students. Thank you so much, and we ask that the competitors stay for photos. So thank you all for being here.